Dear students, today we are going to learn gibberellins, an important phytohormone endogenously secreted in the plant. I am Dr. Alka Inamdr. In Alka's Botany channel, please like, share and subscribe the channel. In this video, we are going to learn introduction of plant hormones, introduction of gibberellins, discovery and chemical nature of gibberellins, distribution of gibberellins in plants, biosynthesis of gibberellins in plants, mechanism of gibberellins action. Last one important point is biochemical or physiological role of gibberellins with practical applications. Plant hormones are the important chemical substances which are synthesized by the plants to regulate the growth in plants, and that's why it is called plant hormones or plant growth regulators. These are produced by plants in very low concentration for the effective results to regulate the physiological plant processes which are going on in the plant. Phytohormones are usually moved within the plant from a site of production to the site of action. Gibberellins are the plant growth hormones which stimulate longitudinal stem growth. These are the group of hormones. Gibberellins were discovered in 1926 by Japanese plant pathologist Kurosawa. The discovery of gibberellins is associated with the cause of rice disease called Buckney, meaning foolish seedlings. It was observed that the seedlings in rice field infected by fungus Gibberella fusiculae were pale and taller. Sivada in 1912 postulated that the cause of Bucknell disease was some chemical secreted by the fungal hyphae in the cells. Yabuta and Hayashi in 1939 were able to isolate two crystalline forms of active material in culture filtrates of Gibberella fusiculae and named them as gibberellin A and gibberellin B. Later, in 1954, Drine isolated and chemically characterized a pure compound from culture filtrate of Gibberella fusiculae and named it as gibberellic acid. We are going to learn chemical nature of gibberellins. At present, there are near about 70 different gibberellins have been identified and named as gibberellic acid 1, that is GA1, GA2, GA3, GA4, like GA70, etc. Original gibberellic acid is GA3, that is a gibberellic acid 3. All gibberellins have similar structure with 19 or 20 carbon atoms, and basically they are terpenoids with 4 or 5 rings. This skeleton is called gibberellin or gibben skeleton. They are having carboxyl group, so they are acidic in nature. In plants, it is distributed. About 70 different gibberellins are now known. They are extracted from fungi, algae, and many higher plants. As many as 30 different gibberellins are isolated from fungus Gibberella fusiculae. Katsumi in 1969 obtained gibberellic acid 3 in 130 species of gymnosperms and angiosperms. They are present in different organs and tissues like root, shoot, birds, flower apices, fruits, and callus tissues. In the biosynthesis of gibberellins, green plants follow the same path for gibberellin synthesis in a fungus, Gibberella fusiculae. Acetate acts as a precursor for synthesis of gibberellins in plants. Acetate produces acetyl coenzyme A molecule, which after condensation forms mevalonic acid. Later on, by a series of reactions, mevalonic acid produces isopentenyl pyrophosphate, a 5-carbon isoprenoid unit to produce carotenoids, gibberellins, etc. The reaction involves coenzymes and carboxylating enzymes. This is the mechanism of gibberellins. The physiological effects of gibberellic acid are mediated via increased enzyme synthesis and membrane permeability. Because gibberellic acid involves an alternation of nucleic acid directed gene level action. The synthesis of plant enzymes such as alpha and beta amylase, proteases, ribonucleases, and nitroreductase is induced by gibberellic acid. It is found that level of newly formed RNA increases due to activation or depression of DNA by one or more genes by effect of gibberellic acid. This favors synthesis of new mRNAs, which produces new enzymes resulting in growth. Gibberellic acid interacts with DNA and changes its properties to lead the growth. Gibberellic acid also affects via increased permeability of the cellular and organelles membrane. And last one point is biochemical role and practical applications of gibberellins. The first one application is stem elongation. Gibberellins stimulate extensive stem and leaf elongation in the intact plants. Practically, gibberellic acid can be applied to sugarcane sets for the stem elongation, which will increase yield and sugar percentage. Thus, farmers can obtain more money. Second one is reversal of genetic dwarfism. The most striking effect of gibberellin is conversion of genetically dwarf plants of normal height. Rosette, sugar beet, dwarf pea, and corn varieties show marked that is a longitudinal growth and attain normal height due to gibberellic acid treatment. Original dwarf plants have low gibberellic acid concentration and external applications will stimulate cell growth due to activation of subapical meristems resulting into increased growth. To increase the height of the genetically dwarf plants, these are useful for plant breeding programs. Third one application is bolting and flowering. Many herbaceous biennial or annual plants from rosette of leaves due to stem with short internodes during first season of growth and during next year they develop long shoot with elongated internodes on which flowers are produced. This phenomenon is known as bolting, which is induced due to winter cold. Practically, it is used by treating biennial plants of economic importance with gibberellic acid. Bolting can be induced and seeds can be obtained in one season or year instead of 2 years. Second one is promotion of seed germination. Certain seeds fail to germinate and remain dormant unless there is a germination factors are available. Gibberellic acid is applied to such dormant seeds like barley, lettuce, tobacco. They start germination without any specific treatment. Due to effect of externally supplied gibberellic acid, hydrolysis and transport of stored food material starts towards embryo leading germination. Dormancy in economically important seeds can be broken and germination percentage can be increased practically due to the gibberellic acid. Fifth one is breaking of dormancy in buds. Gibberellins overcome dormancy in buds in many evergreen, deciduous trees and shrubs. The bud dormancy in winter can be broken by external applications of gibberellic acid. J is also breaks dormancy in potato tubers. Practically, it is used. The process of growth can be induced using gibberellin solution at proper concentration. Next one is substituting as a cold treatment and flowering induction. Normally, winter varieties of plant requires exposure to low temperature during winter season before they are exposed to appropriate light condition. In such plants, flowering can be induced by gibberellic acid application without cold treatment. Winter variety plants of economic importance can be made to flower during any season of application of gibberellic acid. Example, radish, xanthium species, etc. Next one practical application is reversion of sex expression. Gibberellic acid application can change sex expression in some plants. The plants with unisexual flowers, when treated with gibberellic acid, shows increase in number of opposite sex flowers. Example, in Cucumis sativus, cannabis species, male flowers are converted to female flowers and their number is increased, due to which increase in number of fruits per plant can be obtained. Next one important practical application or biochemical role is of parthenocarpy. The formation of seedless fruits without fertilization can be induced by gibberellin treatment. Practically, in agriculture, this is widely used. Gibberellic acid can be successfully used for parthenocarpic fruit development in pome, stone fruits, seedless fleshy tomatoes, and large-sized grapes, and now produced by farmers by gibberellic treatment on commercial scale. The grape varieties like Thompson seedless, Sonaka, Sharad seedless are treated with gibberellic acid 3 for better size and yield. For improving size, color, quality of fruits like apple, tomatoes are treated with gibberellic acid 3 in the form of sprays. And last one practical application is denosynthesis of alpha amylase. Gibberellic acid induces DNO, that is a newly synthesis of alpha amylase enzyme in aleron layer of endosperm in many cereals during germination. This enzyme causes starch hydrolysis to produce simple sugars, which are translocated to embryo to provide energy during germination. Similarly, enzymes like alpha amylase, ribonucleases, proteases, nitrate reductase are also induced by gibberellins. So all these are biochemical role or practical applications of gibberellic acid which are widely used. Thank you. Thank you very much for observing this video.